dear participant. Once again, I apologize for the technical delay in today's session. Today we have Dr. Dhanpal Chauhan, Associate Professor in Microbiology. He's from the ACS College, Gangakhed, which is affiliated to the SITM University, Nanded. And he will have a deliberation on the microbial metabolism in day 7 of this continued classroom teaching program. The session is live from our YouTube channel, DBC Media Center, as well as in a virtual platform of the Zoom. So I handed over this session to our resource person, Dr. Dhanpal Chauhan, sir, to proceed for his this session. Dr. Chauhan, sir, the session is over to you.
Thank you, sir.
Yeah.
I'm very much thankful to DB College and Dr. Chauhan, sir, for allow me to interact with you, dear students. So during this COVID pandemic, so college are offline, on online mode, and there is a very restriction to the students. Many of you might have belonging to rural areas where the network is not possible, but those who are in this class, they will get little bit benefit from this lecture. So I am once again thank to Dr. Chauhan sir and his team, especially Dr. Nagesh Dole. He is my colleague also. So dear student, metabolism, we are going to deal with the metabolism, so especially related with microorganism. So you all are aware about the metabolism. So if there is no metabolism, a cell would not be survive. So we cannot expect the metabolism from dead cell. So every living cell, if it passes the metabolic activity, then the cell can reproduce and perform their many activities like their locomotions, then their digestions. All those things are possible if cell is living. So all you are aware about the types of cell. So actually there are two types of cell, a prokaryotic cell and eukaryotic cell. And one more cell is predicted, that is mesokaryotic cell also. So dear student, this cells actually works on the principle of thermodynamics. So thermodynamics means the study of energy. So there are many kinds of energy available in the nature, and the thermodynamics is the transformation of energy from one form to another form. So I'm not going to teach you that thermodynamics, you will get the confused. So our biological cell is also a system, and it just transforming the energy. You know the laws of thermodynamics, that energy cannot be created and cannot be destroyed, just it going to be transformed from one form to another form. So when we think about any living cell and its metabolism, so we can define metabolism as sum of all the biochemical reactions occurs in a cell, and that involves the energy with a variety of enzymes and proteins. So these variety of enzyme and proteins are responsible for the flow of energy in a cell. If there is no enzyme, we could not expect the life without the enzyme. So when we talk about the metabolism, so metabolism is not just a simple topic to be taught in one lecture. So in short, this 1 hour lecture, I will try to make your concept clear, why metabolism is essential. So why we need to study metabolism. So in biological system, you know the basic energetic molecule is ATP, but there are so many other energetic molecules. But we in third year generally talking about the ATP molecule. There are other molecule which contain energy. So ATP is a universal currency of cell market. So why I put here the cell market? So when we go into the market, without the money, we cannot buy anything. So in the same way, without ATP, cell cannot do anything, any activity of its. As like currency, without money, no one asks you who are you. If you have the money, everybody will come to you, everybody will stick to you. In the same way, ATP is also a molecule without which the cell cannot perform its function. So metabolism, as I said, the sum of all biological reactions occurs in living cell. And this sum of biochemical reactions are of two types, that is catabolism and anabolism. So catabolism and anabolism are the two, these two reactions. Catabolism means breakdown of food molecule which actually contain the energy. So this energy we get it from plants. As a human being, we get it from plants. That is photosynthetic process, and some bacteria, that is chemotrophic bacteria, they also obtain their energy by mode of autotrophic means. So this kind of energy that is stored in food molecule, when it enters in a cell, these molecules are break down, and from that molecule energy is conserved. When energy is conserved, this energy is used for the anabolism. So catabolism means a breakdown of large complex molecule into simpler one, and from this simpler molecule, cell builds its own other building blocks. Means when one cell divide into two cell, this cell exactly need all the raw material. अभी मैं इसको ऐसे ऐसे समझाऊंगा आपको कि अगर दो भाई है और एक भाई अपने घर से अलग हो रहा है, तो पिताजी का व्हाट इज द रोल ऑफ फादर टू मेक द न्यू होम. अगर न्यू होम बनाना है तो ऑल द रॉ मटेरियल, जो ब्रिक्स लगेंगे, सीमेंट लगेगा, एवरीथिंग एंड प्लॉट. सो एग्जैक्टली द सेम थिंग्स नीड टू बिल्ड द न्यू सेल. अभी नया एक सेल से दो सेल बनाने के लिए जो भी रॉ मटेरियल्स है, राइट फ्रॉम डीएनए टू ऑल अदर कंटेंट्स, दे मस्ट बी नीड इन half amount. So this half amount of nutrient, where from it comes? It comes from the food, means energy is flowing. So what are the biomolecules? You know, there is protein, carbohydrates, lipids, nucleic acid. These are the biomolecules. And these biomolecules generally comes from food. So when these biomolecules are broken down, they broken down into simpler one, energy conserved in that biomolecules is released. When this energy is released, this energy used to make biosynthetic reactions. So for example, energy is generated at the metabolites, means cell synthesize their own metabolites. अभी डीएनए को सिंथेसाइज करना है, तो न्यूक्लिक एसिड के जो पेंटोज शुगर है, नाइट्रोजन बेसेस है, ये जो भी असेंबलिंग मॉलिक्यूल्स है उसको मेटाबोलाइट्स बोलते हैं. These metabolite molecules are synthesized by cell using their own pathways. So catabolism generally generate the ATP molecule, as well as they also synthesize the precursors molecule, and these precursors molecule builds a new molecule in the cell, that is catabolism. So anabolism means whatever the precursors molecules are synthesized by catabolic pathways, these catabolic metabolites then assembled to form new molecule. It may be protein, carbohydrates, cell, other materials you can say. So this is the mechanism of anabolism. Anabolism generally use ATP, which is formed by catabolic pathways. So an ATP formation is occur in cell by three mode according to the cell. It is substrate level phosphorylation and oxidative phosphorylation, photophosphorylation. So by these three methods, ATP is synthesized in a cell, and this synthesized ATP is used in the anabolism, means synthesis of a large molecule like DNA synthesis, protein synthesis, lipid synthesis, and so many other biomolecules are synthesized. So during metabolism, near about 15 metabolites are synthesized. So when any cell undergo metabolism, means it performs certain functions, energy comes, energy flows, catabolism occurs, anabolism occurs. These reactions are mainly based on the enzymes. As I said before, without enzyme, we cannot expect the life. So that's why it is very essential to bird eye view on enzymes. So all you are aware about the enzymes. Enzymes are biocatalysts, and that accelerate the rate of reaction by lowering the energy of activation. So when energy of activation is lowered down, the speed of reaction is occurred. So catalysis is very important. So you can understand this by putting one example. Your teachers are your good catalyst. So you are initially lazy, you are not interested in any subject, but if you attend the lecture of your teacher, some idea will get cleared. So you are automatically accelerate to do more study. So it means your teacher is your so in biological system, enzymes are catalyst, and cell is working on its physical parameters, biochemical parameters like temperature, pH, and pressure. So all these parameters at which the cell can function. So that now we define enzymes are biological catalysts. They accelerate the rate of biochemical reactions by lowering the activation of energy. So that is a definition of enzyme. So without enzyme, no cell could be possible, as I said before. So enzyme increases the reaction rate by 10 raised to 6 to 10 raised to 9, 12 times over the uncatalyzed reaction. So because of the enzyme, we can easily digest the food within 24 hours. But if there is no enzyme, the same food we unable to digest for many years. Imagine if there are certain animals who are dying every day on a roadside, you might have observed. So they are degraded within a week. Why this is so? Because microorganism deposited on the body of that animal, we can say carcasses, and microbial activities decompose that body within a few days. If there were no enzymes, it is not possible to degrade that animal for many years, and you might have observed today, if there is no enzyme, everything will be undegradable. So because of the enzyme, it is possible, and you can see the reaction rate increased by enzyme catalyzed reaction from 10 raised to 6 to 10 raised to 12 times. So imagine the enzymes have a powerful biomolecules which carry out the metabolism. So I'm going very short because there is time bound session this is. So enzymes can be named by adding ASC to the substrate whom they are active. So example, you can take urease or tyrosinase. So ASC is a suffix which add to the substrate and the enzymes are nominated. But this make many confusion among the scientific community. Then one more task has been taken by International Union of Biochemistry, IUB, and that decided to precise the name of the molecule. And then that IUBC classified the enzyme into 6 classes. On that basis, these classes are oxidoreductase, transferases, hydrolases, lyase, isomerase, and ligases. So these are the 6 classes. Remember, one more class have been invented, but it is not included in this present curriculum. So this IUB naming system of nomenclature, they nominated the enzyme, and they put one commission, that is enzyme commission code is given to any enzyme. So according to these 6 classes, the first letter should be a class, second letter could be subclass, third one be subclass, and the fourth digit which is related with the individual substrate where this reaction is carried out its functional group. So for example, here D-glucose 6-phosphotransferase is the enzyme. According to the enzyme commission, this enzyme is recognized as EC2712. This is the nomenclature. You can see this is the nomenclature of enzyme. So this nomenclature of enzyme, you can see this is the nomenclature, and according to this nomenclature, the enzymes are recognized all over the world. So second letter is class. It may be oxidoreductase. So here name is 2 is given. So 2 means second class is transferases. That's why it is transferases. If instead of this 2, if I put another name, 3, suppose 3712, it will be the hydrolases. So by that way, this 4-letter nomenclature is included to identify precisely the enzyme. Precisely the enzyme can be identified. So this is classification in short. So how enzymes work actually, that is very interesting part. So it will need more time to make you clear of your ideas. So when enzyme react with the substrate, it will form enzyme substrate complex, and that complex leads to break product, and at the end enzyme is going to be free. So when enzyme combine with the substrate, ES complex is formed that determine the velocity of the reaction. So to determine the velocity of reaction, the scientists Michaelis and Mentane, they determine that when at the initial period, if we going to add substrate concentration and the enzyme, as we going to increase the substrate concentration, it which reaches at the peak saturation peak where the enzyme and substrate are equally matches, beyond which if we increasing the substrate concentration, there is no effect on enzyme substrate reaction, and the that point is maximum velocity. So it was unable to determine the exact velocity of the reaction. Then Michaelis-Menten put their equation to find out the velocity and the concentration of the reaction, and they derive an equation that we also known as equation. According to this equation, Vmax S Km plus S. So this is the equation of Michaelis-Menten. Here initial velocity V0 means initial velocity, S means the substrate concentration, Vmax means maximum velocity, Km is the substrate concentration at half Vmax. Here this is the graph which showing that Km means substrate concentration. So that Km is attained when the maximum velocity of reaction is half. So that is what is called as Km. So Km have many significance to determine the efficiency of enzyme. We usually find out the Km of any enzyme On that basis, we can say that enzyme is good or bad. So this Michaelis-Menten equation is transformed into linear form. So this form is unable to find out the exact Km. Then Lineweaver-Brook plot, that we call as double reciprocal plot, that plot is able to find out the Vmax and the Km, and that is very useful to find out the exact Km and Vmax of the enzyme substrate reaction. So second is once the enzyme kinetic is known to us, then enzyme inhibition is very important part of any research industries. So nowadays you are facing the corona pandemic. So many scientists try to find out the enzyme which inhibit different kinds of molecular reactions. So the once the reaction is stopped, then you will get the rate from such toxic product of the enzymatic reaction. So inhibition means enzyme activity is stopped either by any molecules which is designed. So enzyme inhibition is of three types, that is competitive inhibition, non-competitive inhibition, and uncompetitive inhibitions. So enzyme inhibition is very useful to design drugs. अगर आपको एंजाइम का इनहिबिशन पता चल गया and if you are trying to Km and Vmax, definitely you can be upper hand in enzyme biology. So competitive inhibitors. So competitive inhibitors are those inhibitors which compete with the substrate to bind for the enzymes. अभी जो competitive inhibitors है, it can compete with the original substrate for the active site of the enzyme. So here on your screen, enzyme plus substrate ES complex is formed. Then enzyme is free, at the end product is formed. If inhibitor is bind, it will form enzyme and inhibitor complex, and then the reaction is inhibited. So competitive inhibition can be reversible. So it has many medical importance. Generally, those person who consumes methanol. So methanol is toxic. It may damage your eye permanently, or it may cause your liver to coagulate like egg. So methanol poisoning can be reversed with ethanol. So if you treat the patient who are consume methanol, we add ethanol to it, and the competitive inhibition is reversible, where the application is in medical science. Non-competitive inhibitions. So non-competitive inhibition is in which these inhibitors is bind with either ES complex or it bind with the enzymes. So this non-competitive inhibition is binding with ES complex or it bind with the enzyme, and will definitely affect the reaction. So generally this competitive, non-competitive and uncompetitive inhibitors, they are reversible. So uncompetitive inhibitors, it is not compete with the substrate, it is directly bind with the ES complex. It bind with the ES complex. So when it bind with the ES complex, ultimately it affect both Km and Vmax. So this is competitive, non-competitive and uncompetitive inhibitors. So now with a short description of enzymes. So enzymology is very big area. So that need two more lectures to make your concept clear. But as you are student of third year, I may hope you might have learned this enzyme biology in your classes also. So now we discuss type of respiration, actually metabolism. So as I said before, that metabolism is sum of catabolism and anabolism. So when we talk about the respiration, you might have come across with one things that respiration means taking oxygen inside and releasing carbon dioxide outside. So this we can say this is respiration, but this is actually not respiration. Respiration means in presence of oxygen, involvement of oxygen to break down the biological molecules, which food molecule we can say. So in aerobic respiration, ATP generated from food, that means ATP is conserved with the involvement of molecular oxygen. So where is molecular oxygen present, we will discuss later on. Aerobic respiration means energy is conserved with involvement of molecular oxygen, is called as aerobic respiration. Anaerobic, same method, energy is conserved but the molecular oxygen is not involved, means electron which are generated. So I will tell you when we discuss aerobic respiration where is electron, where from electron comes, and who is going to accept the electron. So energy is conserved and final electron acceptor is nitrate, sulfate, carbonate, but not molecular oxygen. So in aerobic respiration means aerobic respiration in which the final electron acceptor is molecular oxygen. In anaerobic respiration, final electron acceptor is not oxygen. Instead of oxygen, there is a nitrate, there is a sulfate or carbonate, but not molecular oxygen. So fermentation is one type of respiration for the survival of organism. So fermentation is one of the methods by which the organism is surviving. So electrons are generated. If that electron is not accepted by the final electron acceptor, cell will definitely dwindles. So that's why fermentation is one of the methods for the surviving. This strategy used by some microorganism to survive. So fermentation is in which the final electron acceptor is an organic compound. So organic compound itself acts as an electron acceptor or electron donor. So now the idea is going to be clear. Aerobic respiration in which final electron acceptor is oxygen. In anaerobic respiration, final electron acceptor is not oxygen. Instead of oxygen, there is a nitrate, sulfate, or carbonate. And in fermentation, final electron acceptor is organic compound. So organic compound is final electron acceptor as well as it is electron donor, right? So now we are going to discuss about the respiration, aerobic respiration. So aerobic respiration, initially you might have observed here equation which you have learned in your previous or basic classes, that glucose in presence of oxygen, it will leads carbon dioxide, water and ATP. So glucose is a molecule, that is food molecule. When enters in a cell, cellular respiration, when glucose is entered, what kind of episode occur inside the cell so that it can able to generate carbon dioxide and water along with energy conserved in the form of ATP. So if you see at this slide, sir, ये slide दिख रहा है क्या मुझे? Students, I am going to audible or it is one-way process?
No, sir. Your slide is not visible, but you're audible, sir.
Slide is not visible.
No, sir.
No slide is visible.
I think the screen is going to be black there. Previously, there was a slide, but right now it is not there.
Slide is not here. Okay, so I think my slide is.
No, earlier it was there.
Then now it is possible. It is paused now, I think.
Share once again, sir.
Now?
Yeah, it's coming, sir.
Okay, okay. Student, see here, you will find this slide. This is aerobic respiration. So when you see this aerobic respiration, glucose is the molecule. Oh, once again, it is paused, na, sir?
Sorry, sir.
Slide is again disabled, na?
No, sir. It is there, sir. It is on the screen. Just move it up.
I think. Okay, you see this slide. I think it is visible, na?
Yes, sir. Yes, sir.
Okay. In aerobic respiration, student, see here, this is glucose molecule in presence of oxygen. Inside the cell, you will find that carbon dioxide, water and ATP is generated. So when this whole episode occur inside the cell, behind this whole episode, cell perform many kinds of activities to generate this CO2 and H2O. It is not easily generated inside the cell. So cell need to go many rounds of its enzymatic reaction so that we can see this CO2 and H2O along with ATP conservation. So if you see at a catabolic reaction, here first the glycolysis that we can call as the EMP pathway. So glycolysis or EMP pathway, it occurs inside the cell cytoplasm. When cell get a glucose inside it, all the enzymes of glucose breakdowns are located in cytoplasm. So cell cytoplasm is full with enzyme of glycolysis. So glucose is broken down into pyruvic acid. So glycolysis is occur in presence of oxygen or it occur in absence of oxygen. So glycolysis occur in those conditions where there is a limited oxygen or oxygen. So that's why glycolysis occurs in both conditions. So glycolysis, that is EMP pathway, occurs in microorganism as well as in human and plant cell. So when glycolysis occurs, enzymatic breakdown occur inside the cytoplasm and glucose is able to break down with with the action of many enzymes, and finally it leads to form the pyruvate. You can see here pyruvate is formed from glucose. It forms the pyruvate, and this pyruvate is ready now. So when glycolysis occurs, glycolysis yields kinds of molecules. So one is electrons are generated via NADH. So this NADH is a reducing power, you can say. How it is? Because NADH means hydrogen molecule is released when glucose is break down. This hydrogen molecule is released. If this hydrogen molecule is not taken up by the cell, cell could not survive. So naturally inside the cell, these NADs are present. So these NAD plus get the hydrogen and reduce to NADH. So cell is managed to survive. And when glycolysis is carried out, pyruvate is formed. The end product of glycolysis is pyruvate, and NADH is generated. and ATP is formed, means energy is conserved here. But the mode of ATP formation in glycolysis is substrate level phosphorylation. So substrate level phosphorylation means the way one substrate is converted into another substrate, then a phosphate molecule is transferred to ADP to form ATP. So these all reactions occur inside the cell cytosol, means cytoplasm of cell, where glycolysis is occur. So when glycolysis occurs, it generates ATP and it generates NADH molecules. So ATP here generated is usable by the cell, used by the cell, and NADH, it has energy. NADH has the energy. This NADH containing energy is generated in electron transport chain. So that chain is we discuss in aerobic respiration. So now when pyruvate is formed, pyruvate has many fates. Either it enters in aerobic respiration, anaerobic respiration, or fermentation. So pyruvate is the key metabolites of a cell. So pyruvate is a key metabolites of a cell. So here I am going to share my next screen, but it is not visible, sir.
Sir, do one thing. Reshare your slides, sir.
Reshare, okay.
Shall I share, sir, from my side?
Okay, sir. You can also share, no problem. Now it is visible now, I think.
Yes, sir. Yes, sir. Yes, sir.
Oh, okay. So now aerobic respiration, right here, when pyruvate is formed, that pyruvate enters in a Krebs cycle. So you know that Krebs cycle is occurred in mitochondria, and student, you might have heard mitochondria is called as a powerhouse of the cell. Why it is called as powerhouse of the cell? Because this pyruvate, when enters, this pyruvate has too much energy. When this pyruvate is enters in mitochondria, then Krebs cycle is start. So pyruvate is first decarboxylated, form into acetyl CoA, and that acetyl Krebs cycle. So Krebs cycle is located in mitochondria, where electron transport chain is also located. If organism is aerobic, then this pyruvate is enters in mitochondria. So aerobic organism has mitochondria. Some bacteria are also aerobic, they do not have mitochondria, but Krebs cycle and electron transport chain is there. So where does this Krebs cycle and electron transport located in bacteria? The answer is that is located in a plasma membrane of bacteria, because prokaryote lacks mitochondria. That's why the Krebs cycle and electron transport chain is located in their plasma membrane. So once the pyruvate is formed in anaerobic condition, it enters into a Krebs cycle. When it enters in Krebs cycle, energy which is present in pyruvate, it is again generated in Krebs cycle. So Krebs cycle ATP, and that ATP is formed by substrate level phosphorylation again. So from the Krebs cycle, you find that there is a generation of ATP by substrate level phosphorylation, and there is a generation of NADH, 2 NADH molecule and 1 FADH molecule is generated. So NADH molecule generated in glycolysis, it enters in electron transport chain, And when pyruvate enters in Krebs cycle, it also generates the NAD and FAD. That all NADH and FADH, which is generated glycolysis and Krebs cycle, they finally reaches to the electron transport chain. This electron transport chain is located in a mitochondrial inner membrane, where the ATP is generated by oxidative phosphorylation. And you might have known that one NADH molecule can generate three molecules of ATP. So majority molecule generated by these NADH when they enter in electron transport chain. That's why mitochondria is called as powerhouse of the cell. So if you see at this link, glycolysis, Krebs cycle, and electron transport chain. So this is completely aerobic respiration. This is complete the aerobic respiration. When glucose molecule is entered, you will find that and glycolysis occurs. Then pyruvate is entered in Krebs cycle, and Krebs cycle and glycolysis generate large number of metabolites, that many amino acids are synthesized from the intermediate of Krebs cycles. So that we will discuss. That's why Krebs cycle is also called as amphibolic pathways. So you can see here the catabolic pathways of glucose breakdown. So glucose is converted into glucose 6-phosphate. That glucose 6-phosphate is fructose 6-phosphate. Then fructose 1,6-bisphosphate. Then 3-carbon compound, that oxyacetone phosphate and glyceraldehyde phosphate. So overall, when you see this, the ultimate aim of glycolysis is to generate the pyruvate. So 2 ATP molecule is generated, and 2 pyruvic acid molecule is generated from simple glucose in glycolytic pathways. So many intermediate of glycolytic pathways are used for generation of metabolites. So now when pyruvic acid is formed, oxidative decarboxylation occurs, and this is the picture of citric acid cycle, which is on the screen. You can see citric acid cycle. So Krebs was the scientist who designed the structure of this citric acid cycle. We can call as a tricarboxylic acid cycle also. So when that acetyl CoA, which is formed from pyruvate, means decarboxylation of pyruvate occurs, and then it leads to form acetyl CoA. This is the reaction before it enters into Krebs cycle. So when acetyl CoA is enters in a cycle, it reacts with oxaloacetate in the presence of enzyme synthase, citrate synthase, you can say, and then the whole reactions is occur sequentially. Oxalate to citrate, cisacotinase, isocitrate, oxalosuccinate, alpha ketoglutarate, then succinyl CoA, succinate, fumarate, malate. So these are the sequence, and these are the enzymes which is shown here in this figure. So what is the importance of this citric acid cycle? So importance of citric acid cycle is that many amino acids are synthesized from these precursors. For example, alpha ketoglutarate, then succinyl CoA, then oxalosuccinate, fumarate. These are the intermediate molecule from which the cell precursors molecule are synthesized. That's why this citric acid cycle has importance. This is the hub of aerobic respiration. If there is no citric acid cycle, then no aerobic respiration is possible. That's why citric acid cycle is also called as a hub of aerobic respiration. There is a modification, those bacteria which microorganism where the citric acid cycle is not there, then glycosylate cycle is there, which also produce the same intermediate molecules. So citric acid cycle is bypassed in glycosylate cycle, that we will discuss later. So now the previous figure which I showed you, that NADH molecule is generated, that enters in Krebs cycle. Then from Krebs cycle they are enters into electron transport chain. So in electron transport chain cycle, this electron transport chain present in mitochondria, and as I said, it is present in bacteria also in plasma membrane. So when this NADH molecules enters in electron transport chain, so electron function, this cycle separate the electron and proton from hydrogen molecules. You know, hydrogen molecule has one proton and one electron. So when this hydrogen molecule enters in electron transport chain, the component chains are very specific to separate the hydrogen from proton. That's why it is very essential how it is naturally present, that the machine, cellular machine, separate the proton from hydrogen, and this is carried out by electron transport chain. So there are certain molecules which transport the electron, for example cytochrome, then quinone molecules. These have very specific to separate hydrogen from electron. So when separated, you can see here there are the complex, first one complex 1, complex 2 and complex 3 in bacteria, and in eukaryotic there is slight bit difference. So eukaryotic electron transport chain is not branched, whereas the prokaryotic electron transport chain is branched, and it is separated in branched electron transport chain present in bacteria. It allowed them to survive in different environmental conditions, so that the final electron acceptor is achieved. So these electron transport components, they separate the electrons and they transport the electrons. Along with when this proton are separated, proton are channelized out to the outside of the membrane. When this NADH enters in electron transport chain, so see here NADH H plus, then NAD is again oxidized. So this hydrogen molecule is entered. So this hydrogen molecule enters, it then electron is transported to the quinone, and then it is going to be cytochrome, and then it is going to cytochrome C, then cytochrome A3, and finally it reaches to the oxygen. So there are complex NADH complex, cytochrome B1 complex, cytochrome oxidase complex. So when the electrons which are that hydrogen molecule which are generated in Krebs cycle, generated in glycolysis, now they enters in electron transport chain. When this electron are transported to highly electronegative oxygen, so during this electron transport chain, hydrogen and electrons are going to be separated by the component of electron transport chain. Hydrogen molecules are gaining, proton molecules are gaining outside, and the cytoplasm become more negative and the outer side become more positive. So that make the proton gradient across the membrane because of electron transport chain. So these hydrogen molecules, these proton molecules has the energy. So these protons, H plus ion, are not easily permeable to the cytoplasmic membrane, not easily permeable. Then in electron transport chain, there is one of the enzyme which is present in electron transport chain, definitely in the membrane, that called as ATP synthase. So this ATP synthase molecule is a smallest motor which rotate in this fashion so that the ATP is formed. So these H plus ion which enters outside the membrane, they can channelize through this ATP synthase. When they enter through this ATP synthase, then these electrons are taken up by half O2, and then it leads to form react with the hydrogen molecule, half O2 and the hydrogen molecule means H plus molecule through this ATP synthase. When they enters, then ADP is converted into ATP. ADP is converted into ATP by the H plus ion, then ATP is formed. This kind of formation of ATP from H plus ion is called as oxidative phosphorylation. So oxidative phosphorylation means ATP is generated from H plus ion. So this H plus ion, where from it comes? It comes from glycolysis and Krebs cycle, which then enters into electron transport chain, and then it enters through ATP synthase and ATP is formed. Then final electron, which final electron means electron released from glycolysis and Krebs cycles, they finally reaches to the oxygen. That's why that is aerobic respiration. So as I said before, aerobic respiration is when the final electron acceptor is oxygen, then that is called as aerobic respiration. When aerobic respiration occurs, glycolysis, Krebs cycle, and electron transport chain where the ATP is formed, then oxygen combine with electron and to form H2O. So the initial equation, glucose plus oxygen, it form CO2 and H2O. So where is CO2 molecules? When Krebs cycle, you can see Krebs cycle. In Krebs cycle here, the CO2 molecule is generated. In Krebs cycle, 2 CO2 molecules are generated and one in oxidative decarboxylation. So then this equation is completed. So when glucose is enters, it in presence of oxygen, it leads to form CO2 and H2O. So here H2O is formed and CO2 in Krebs cycle you can see. Then ATP, the equation was ATP. Here you can see, here I mentioned earlier this glucose plus oxygen, it leads to form oxygen plus water plus ATP. So when we go across this, ATP is formed in Krebs cycle also, in glycolysis also, and in electron transport chain also. This equation is complete when we study glycolysis, Krebs cycle, and electron transport chain, where our equation is fulfilled. So anaerobic respiration. So as I said, in anaerobic respiration, final electron is not oxygen, but instead of oxygen, the final electron is nitrate, sulfate, fumarate, and etc. So anaerobic respiration, where the energy is also conserved, and final electron acceptors are these molecules that we discuss later. So fermentation. So pyruvate is the key metabolites. If the cell is anaerobic, or the bacteria which utilize fermentation pathways, this pyruvate is broken down to form large number of molecules. So whatever the NAD molecule release in glycolysis, they must be released. They must be released, and when they released, this must be gained. As I said, fermentation is a process in which organic molecule acts as electron acceptor as well as electron donor. So when pyruvate is organic molecule, when it get the reduced, when it reduced by gaining the electron, then pyruvate can it may be acetic acid, propionic acid, and butyric acid. So many kind of fermentation will be formed, ferment product will be formed when pyruvate is enters in a fermentation pathway. So dear student, now it is not possible to explain each and everything within this short period of time. So I conclude with this session. If anybody want to ask me question regarding this aerobic respiration, so when I get the time, I will I will take your aerobic respiration, anaerobic respiration, and fermentation in greater detail. So I am once again very thankful to Dr. Chavan sir. He is a good friend of mine. Dr. Dhole sir is also a good friend of mine. I thank you all by inviting me to my short talk on metabolism, sir. Over to Dr. Chavan sir.
Sir, yeah, thank you. Thank you very much, sir, for the nice presentation in this countrywide classroom teaching program. Sir, will you stop sharing this slide? John, sir? Yeah, okay, thank you. Thank you. Thank you, sir, for a nice presentation, and hoping that you will be available once again in this countrywide classroom teaching program. Earlier, we had done this program for the almost 1 month in April 2020, and around there are 58 lectures has been recorded, or the e-content has been developed in this session. In this countrywide classroom earlier countrywide classroom teaching program, and hoping with that in this countrywide classroom teaching program, we also recorded around 50 plus e-contents for our students of the university, home university, as well as the other universities, with you people supports only. So you will be available for this countrywide classroom teaching program. I handed over this session for the vote of thanks to the Dr. Nagesh Dhole, sir.
Thank you, sir. A great lecture given by Dr. Dhanpal Chavan, sir. He has described the microbial metabolism in very similar, simple, and many students they can understand very easily. Sir highlighted many topics: introduction to cellular functions, how energy generates, how energy utilized, how oxidative phosphorylation takes place, substrate phosphorylations. So I have created a basic knowledge, and after that he has started the lecture. So also described about the enzymes, what is the catalyst, what are the classification of enzymes, then what is enzyme commissions and their types, different types of the enzyme inhibitions, all parts I have taken. After that, so briefly describe about the aerobic, then anaerobic respiration, and finally fermentation. Thank you, sir, for being a part of this countrywide classroom. Once again, thank you.
Okay, sir.
Chaman, sir.
Okay, sir. Thank you very much.
Thank you very much, sir. Hoping that you will be available once again in this countrywide classroom teaching program. I ended this session.
Okay, sir. Thank you very much.
Yeah, dear participant, tomorrow we will be tuned at sharp 2 noon o'clock. Thank you. Thank you very much.